This is the lecture for the muscular system, and we're getting into our last section of material, our last unit for the semester. So we have muscular system this week. This coming week we have the urinary system, so excretion and homeostasis. After that we have reproduction and inheritance, and then that's it for the material that we're covering in lecture. So at that point you'll have some time to work on your term papers, focus on lab, you'll have fall break, and you'll really have a lot of opportunities to focus on studying for the final exam. So remember, the weeks or the modules are kind of arranged by week, and so every time we have an exam, that week has your grades for the Scantron and the written exam, but it also has a review resource. So kind of the most missed questions, some pointers on how to study for those, and an indication of the material that you'll see again on the final exam. I have that resource done for the second lecture exam. I'm just waiting on some people who had documented reasons for taking the exam a little bit later to finish taking it, and then I can post it to Canvas. I don't want to post the most missed questions until everyone takes the exam. So hopefully that will be posted by Tuesday at the absolute latest. I'll try to get it to you guys soon. Everything is graded if you've already taken the exam, and it's on Canvas, and you will get your graded written exams with feedback on Friday, since I had them in office hours, but nobody came to office hours because you guys had your lab exam. So for this learning unit, the muscular system, these are the focus bullet points that are on Canvas, so make sure you scan those before you go through the lecture slides. So we're talking about muscle tissue today. It's just one chapter, but it's quite a bit of material. So we're putting together a lot of ideas from the nervous system, which we finished talking about in the last kind of unit or section of the course. We're going to start out by briefly reviewing anatomy, and we're really focusing a lot on skeletal muscle. So towards the end of this lecture, we'll review cardiac muscle, smooth muscle, and kind of muscle development and regeneration, but the bulk of this will be about signaling and contraction in skeletal muscle. So we'll talk about skeletal muscle specifically, muscle fiber contraction and relaxation, nervous system control of muscle tension, and the effects of exercise on physiology, specifically with muscle performance. So before we get much further, I wanted to talk briefly about muscular dystrophy. This was what the reflection was on this week. And when we see that term muscular dystrophy, it's a very broad term for several types of muscle diseases. So basically, dys means bad or not working too well, and troph has to do with kind of nutrition. So dystrophy is kind of a malnourished or malnutrition appearance of muscles. So it's related to increased weakening and the breakdown of skeletal muscles over time. So we contrast this with things like neuropathy, which have to do with the nervous system being the issue. Remember that the nervous system and skeletal muscles are very closely linked. The skeletal muscles are dependent on the nervous system for signaling and control, and so we try to identify which of those is the problem. In the case of muscular dystrophy, it's the muscles that are not working. And one of the most common types of muscular dystrophy is Duchenne muscular dystrophy, or DMD. It's very common as far as muscular dystrophies go, and the way that it happens is there is a gene called DMD that codes for a protein called dystrophin. Remember, proteins often end in IN, and dystrophin is responsible for binding between these things called thin filaments, which are made up of actin primarily, and the sarcolemma, which is the plasma membrane of the muscle fibers. So structural integrity is very important. If you don't have that binding secure, then you end up with your muscle contractions tearing that plasma membrane. You have calcium ion influx, which leads to a lot of problems, as well as cell damage and muscle degeneration. So another interesting thing about this is because the gene that codes for dystrophin is located on the X chromosome, and because it's a recessive condition, it's more common in people who are chromosomal males, so it's X-linked. And it gets progressively worse over time. It kind of emerges when you're a couple years old, and then there's progression with age. So this is an individual who has this Duchenne muscular dystrophy. Okay, so just a kind of overview of muscle tissues, brief review from anatomy. All muscles, regardless of whether they're skeletal, cardiac, or smooth, share some features in common. So actin, those thin filaments, and myosin that makes up those thick filaments are very important for this process of contraction. Remember that that IN ending tells you that these are proteins. So actin and myosin are going to be very important contractile proteins. We're going to talk about different types of proteins that are found in muscles. Actin and myosin are the consistent ones. Muscle fibers and muscle organs require tons of ATP. We'll talk about different ways that they obtain that ATP, but one really important way is through aerobic respiration with mitochondria. So a lot of these muscle fibers have tons of mitochondria packed into them. Muscle fibers also relax when calcium is removed. Calcium is very important for initiating contraction. We'll talk about the role it plays there. So calcium and ATP are both very important for contraction, and when calcium is removed, that causes relaxation of muscle fibers. Some other features that muscles share in common are this idea of excitability. Remember that cardiac tissue, cardiac muscle, has this property of autorhythmicity. It has the ability to set its own kind of pace and generate its own action potentials. And so muscle in general can receive action potentials, and it's excitable. It can kind of spread that and propagate that. There's a big ion cascade that we'll talk about in a little bit. It's also extensible, so it's able to stretch, but then it snaps back to the way it was, so it's elastic. It returns to its original length when relaxed. And it also has this idea of contractility, where it's able to pull on something. So your muscles are attached, they're inserted and originating in different locations. And so the goal here is to move stuff around, to pull on attachments and shorten, and they use force to accomplish this. So briefly reviewing some stuff from the nervous system in terms of this electrochemical action potential and excitability. Remember, I saw a lot of kind of confusion about the idea of potential on that, the last essay question from the last exam about the nervous system. So I know a lot of people are still struggling with that, but remember the membrane potential, this idea is kind of like having a battery. You have this potential energy stored up in the form of concentration gradients, and the membrane potential is measured, and it's kind of a consistent number at a state of rest because you have consistent ion concentration gradients. And so in most cells, it's about -70 millivolts at resting potential. And so again, we're thinking about this idea of polarization. So becoming depolarized and then repolarized. So is your energy polarized? Is it stored up, or is it actually kind of changing and being used? So excitability is this idea of the plasma membrane going from polarized, so resting at this negative value, to depolarized, more positive, and sending an electrochemical signal. All those voltage-gated channels open up, that keeps the signal going, and then things get restored back to the way they were, so they repolarize. So they're polarized, they depolarize, and then they repolarize, and then at some point they hyperpolarize, I just wanted to review this graph again because this was something that you needed to have been able to label on that written question, and a lot of people struggled with this question. So please make sure you understand this flow of polarization, then depolarization, then repolarization. Remember, there's several types of muscle tissue. There's skeletal, cardiac, and smooth. Skeletal and cardiac are both striated. Cardiac and smooth have less nuclei per cell. Cardiac is really kind of unique in that it has these openings that allow for really consistent electrical connections between cells. Skeletal is also very dependent on the nervous system, whereas cardiac and smooth receive information from other places as well. So just to reiterate that, muscle types respond to signals differently. Skeletal muscle is completely dependent on signaling from the nervous system. I should say from, not form. Cardiac and smooth muscle are influenced somewhat by the nervous system, but they also respond to hormones as well as local stimuli. Okay, so getting into skeletal muscle and the importance of that. This GIF is from a game called QWOP, Q-W-O-P. I don't know if any of you have played it, but it's kind of silly. You have to try to move this individual around, and they don't have really great stabilization. They have great articulation. They have bones and connective tissue. All of their joints are held together, but it's clearly not stabilized very well with muscles. So skeletal muscles in particular are very important for kind of large-scale movement as well as small constant adjustments. So we'll talk a little bit today about the types of contractions and tone that lead to posture. So without skeletal muscles, you might have bones and joints, but they're not aligned very effectively. Smooth muscle tracts also usually end in skeletal muscles at sphincters for increased control. And then skeletal muscle is involved in homeostasis. It's important for shivering, maintaining body temperature, as well as supporting internal organs. So we know that the skeletal muscle is really dependent on the nervous system, and we classify the relationship between, or the physical kind of spatial relationship between a motor neuron and a muscle fiber as the neuromuscular junction, also called the myoneural junction. You're going to see that prefix myo a lot today, M-Y-O. That means muscle. So whenever you see myo in different settings, like myocardium in the heart muscle, or myometrium, the muscle layers in the uterus, that myo is referring to muscle. So whenever you see it today, just know that it's telling you something about muscle. So the axon terminal of the motor neuron is meeting the muscle fiber, specifically at the motor end plate. So we see the axon terminal here and the synaptic bulb of the motor neuron. We see the motor end plate, which is the specific location of the muscle fiber that is kind of adjacent to that axon terminal and the synaptic bulb. Remember that different neurotransmitters, especially acetylcholine, have to be able to kind of be exocytosed and released and flow across the synaptic cleft, the synapse, and then bind to receptors. They're not directly attached to one another, so there's that space, and the motor end plate and synaptic bulb are kind of the closest you get at the neuromuscular junction. I mentioned acetylcholine, which is abbreviated as ACH. That neurotransmitter in particular is crucial for electrochemical signals in muscles. It's what's triggering muscle action potentials. So when we talk about neurotransmitters today, we're focusing on acetylcholine. It is crucial for muscles. In anatomy, you might have also learned about motor units. So this is, again, kind of expanding on the idea of the neuromuscular junction. A single motor neuron usually connects to several muscle fibers, and fibers are individual muscle cells. And so you might have very small motor units where you have one motor neuron maybe connected to 6 muscle cells, muscle fibers. So that's kind of small movements, fine motor control, like your eye muscles. Larger motor units allow for kind of bigger movements like your thigh and your back muscles with thousands of cells, thousands of fibers connected to a single motor neuron. Recruitment is this idea that you can start with smaller motor units and then add increasingly larger motor units with higher thresholds. So to kind of look at individual muscle fibers and components of those muscle fibers, the sarcomere is the functional unit of muscle fibers. We'll talk about all those different letters and the different structures of the sarcomere in just a moment, but first focusing just on the muscle fiber. Each muscle fiber contains myofibrils, which are made of myofilaments. So fibrils are like the mini fibers inside of the fibers, and filaments are smaller structures inside of those. So the thick filaments are made up of myosin, that contractile protein. The thin filaments are made up of actin. So thick filaments, myosin, thin filaments, actin. Those are both types of myofilaments, which are arranged inside of myofibrils, inside of muscle fibers. Those myofilaments are then what are compartmentalized into functional units called sarcomeres. In each sarcomere, we have both thick filaments and thin filaments. And the zones and kind of bands might change a little bit depending on what stage of contraction they're in. So we'll talk about that in just a moment. Before we get into that, you should know that the muscle fiber is very, very specialized. It has a lot of differentiation, and so we have special terms to refer to different kind of common components of muscle fibers. So the sarcolemma is just a fancy way of saying plasma membrane for muscle fibers. Sarco also kind of indicates that it's muscle, so myo and sarco both should indicate to you that it's muscle. Sarcolemma is the plasma membrane of a muscle fiber. Sarcoplasm is the cytoplasm of muscle fiber, and the sarcoplasmic reticulum, or SR, is a specialized smooth endoplasmic reticulum that is very important for controlling calcium ions. So it stores those calcium ions, it releases them, and it also retrieves them and restores them at the end of contraction. So hanging on to those calcium ions and having them readily available when a certain signal comes through is very important for the SR to accomplish. We also have these T-tubules or transverse tubules that connect the plasma membrane, the sarcolemma, and the sarcoplasmic reticulum and are able to kind of transmit those action potentials across the cell. We have different arrangements. We have something called a triad, which is the T-tubule with the membranes of the sarcoplasmic reticulum on either side, and a myofibril, like we talked about earlier, has actin and myosin. It's a cylindrical structure, and it's actually surrounded by that triad. So it's surrounded by the T-tubule and the sarcoplasmic reticulum. So I mentioned some of these proteins already, but I really want you to keep in mind actin, myosin, tropomyosin, and troponin. Actin and myosin are both contractile proteins. Actin is in thin filaments and myosin is in thick filaments. Tropomyosin is a regulatory protein and troponin is as well. Tropomyosin and troponin are both found on thin filaments. So we'll kind of talk about how different signaling molecules affect the arrangement of those thin filaments and how myosin gets access to those thin filaments. So before, actually you can kind of see that here, so let me kind of point this out. So we have these little actin beads right there. So each of those green circles is actin, and you can see that they have sites on them that are where the myosin actually binds. So on actin, we have a binding site for myosin. On myosin, we have actin binding sites that are going to connect to that actin. So we see the thick filament right here, and it has these myosin heads, which have actin binding sites on them, and then those are what's going to bind to the actin. on the myosin binding sites on the actin. The problem is that tropomyosin covers up those binding sites most of the time, unless you have certain signals coming through that are interacting with troponin. So getting troponin to kind of adjust its structure and move out of the way and allow tropomyosin to move out of the way is going to be key for actin and myosin binding. So I just wanted to point that out on this diagram because it does a good job of showing you all those different structures. But now we're going to focus on the structure of the sarcomere. So remember, sarcomeres are the functional unit of the muscle fiber, and a single sarcomere extends from one Z line, which is located here, to the other Z line. And so those appear as striations on the skeletal muscle fiber. We also have I bands, which are basically areas of thin filaments that are by themselves. They don't overlap at all with the thick filaments. So it's just actin on its own, not overlapping with myosin. We also have A bands, which are the length of a single thick filament. So the length of a single myosin filament. The H zone is the area of that A band, of that myosin thick filament, without any thin filament overlapping it. So it's basically, where you don't have any actin. And then the M line is the middle of the H zone. It's kind of this perpendicular center structure that's formed by cytoskeleton. So that's kind of meaningless on its own, but we can see how the sarcomere structure changes as you contract. So we see that the A band is shortening, the A band is not shortening, it's staying consistent, but the H zone is shortening. We see the Z lines getting closer together, the I band shortening. The A band doesn't shorten because the thick filament doesn't shorten at all, but the other structures are kind of the overlap of actin and myosin, or actin on its own, and so we see those things getting smaller. So during contraction, the structure of the sarcomere changes. The Z lines move closer together, the H zone and I band get smaller, but the A band again remains consistent. So that's what happens during contraction. But in order for this to happen, first the muscle fiber has to fire an action potential. That's called excitation. So there's a coupling, a relationship between contraction and excitation called excitation-contraction coupling. So there's the action potential, there's excitation, and that leads to contraction. Those two things are coupled together. So even though resting potential we usually think of as being around -70, for the sarcolemma it's between -60 and -90 millivolts. That membrane potential changes as protein channels like voltage-gated channels open up. Those are embedded in the membrane, they allow ions to move across, and that helps generate an action potential. So I'll walk you through this process. So this first starts by an action potential coming in along an effector neuron, so a motor neuron, to the neuromuscular junction. So it's coming from the spinal cord along an axon of a motor neuron to the NMJ from the somatic nervous system. So remember, it's not autonomic, it's voluntary, it's somatic, and so we have the signal coming into the muscle fibers. So when that happens, remember acetylcholine is very important. It's going to diffuse across the synaptic cleft and bind to receptors on the motor end plate. Before I go on to the next step, I just wanted to point out that this process is very carefully regulated. So we have an enzyme called acetylcholinesterase, which breaks down, or not breaks down, but breaks down and also regulates acetylcholine, because you don't want acetylcholine floating around and just constantly binding to receptors. You want the effect to happen, and then you want to wait. So just like how in our nervous system we have reuptake molecules and enzymes that come through and manage neurotransmitters, in our neuromuscular relationship, we have acetylcholinesterase, which breaks down and regulates acetylcholine. So after acetylcholine binds to those receptors, it opens channels that release positively charged ions, specifically sodium ions, which are positively charged, and causes depolarization. So remember, depolarization is becoming more positive, and so you have that negative resting. Now you have these positive ions coming in. That's causing depolarization, and the more you have this, the more likely you're able to open voltage-gated channels. So those voltage-gated channels, those voltage-gated sodium channels open, that causes rapid depolarization. So you have this very fast depolarization, just like with our nervous system, that initiates an action potential and excitation, which leads to different ions being released. So that excitation causes the release of calcium ion. So we had the signal being sent from the nervous system, we have acetylcholine, we have sodium influx and that changing the polarization and causing depolarization. Now we have the release of calcium ions from the sarcoplasmic reticulum, from the SR. And remember, we talked about the structure of those thin filaments. We talked about tropomyosin wrapping around and blocking those sites where the myosin would bind. So what's going to happen is the calcium is actually going to interact with the troponin, form a complex, and that moves the tropomyosin out of the way. So calcium binds to troponin. It forces those shielding proteins, the tropomyosin, to move aside. Those binding sites are now available to the myosin heads. So the myosin binding sites on actin are free, and the myosin heads can bind to them. Myosin is able to pull on actin and contract the sarcomere in the muscle. And then when you remove that calcium, it causes relaxation. So getting kind of a little bit more into contraction and relaxation. Contraction really has two fundamental requirements. We just talked about calcium. So calcium ions have to be available to bind to troponin and keep binding sites accessible. You have to have those relationships forming between actin and myosin in order for contraction to occur. Also, you have to have ATP to drive what's called cross-bridge cycling and the continuous pulling of actin strands by myosin. So it's not just a one-time thing. We have to have myosin pulling, kind of doing a strong movement, detaching and retaching, and that whole process is called cross-bridge cycling. So if we didn't have ATP, we wouldn't be able to power that, so ATP is very important. So when you do have these requirements met, the muscle remains contracted to an anatomical limit. So there is a limit to how far you can go, but as long as you have calcium ions and ATP, the muscle can remain contracted. So we'll talk about the importance of calcium very briefly after we talk about relaxation. We'll talk about this idea of cross-bridge cycling, and then we'll think about where muscle fibers get ATP. So we talked about how when you remove calcium ions, that causes relaxation. Relaxation is the end of contraction. So it happens when the muscle fiber runs out of calcium ions or runs out of ATP. Remaining calcium ions get pumped back into the sarcoplasmic reticulum. Remember, that stores calcium ions. That removal causes the tropomyosin to then reshield the binding sites on the actin strands. So it shifts back into place and covers up those binding sites until you get calcium again. So those calcium ions really are crucial for unshielding the binding sites. We talked about how the tropomyosin is covering up those binding sites, and there's the troponin complex. When calcium binds to the troponin, it shifts everything off to the side, so the tropomyosin strands move out of the way. Those binding sites on actin are exposed, and myosin can bind to it. So we classify this or characterize it according to the sliding filament model. It's how we conceptualize the function of the sarcomere, this idea of the filaments kind of sliding past each other, and that is only possible, that connection between actin and myosin and the sliding mechanism between thick and thin filaments is only possible if you have these regulatory proteins in place and you interact with them in some way to allow actin and myosin to bind. So those calcium ions allow the complex to form and everything shift out of the way, and then ATP is what allows it to continue to function. So ATP steps in with this idea of cross-bridge cycling, which we'll get to next. So like I mentioned, the cross-bridge cycling is this process of attaching a strong movement, detaching, and then retaching or recocking. So it's kind of like rowing a boat. You put the oar into the water, you have a power stroke, you pull the oar out of the water, and then you place it back into the water a little bit further up, and then you can do another power stroke and push yourself forward. So that attachment, the oar going into the water, is the myosin heads attaching to actin at those binding sites. When this happens, adenosine diphosphate, ADP, and an inorganic phosphate are attached. So that inorganic phosphate gets released. The myosin attaches really strongly to actin when that inorganic phosphate is released, and it's able to pull about 10 nanometers. So that's the power stroke. But then a new ATP comes in and binds, and that causes the myosin heads to detach from that binding site from actin. And then myosin has the ability to undergo ATP hydrolysis. It has ATP-ASE. So ATP is hydrolyzed by this enzyme in the myosin, and then that is able to position the myosin head a little bit more for movement. So it's this recocking. So you have attachment, power stroke, detach, and retach. So this is just kind of another visual of that process. We have the cross-bridge formation, the attachment, we have the power stroke, we have the detachment, and we have the getting it ready again. So remember, we have to keep getting sources of ATP to continue this cycle. ATP comes in and it gets broken down by ATP hydrolysis, by the ATP-ASE from myosin, and you have to keep getting ATP for that cycle to continue. So kind of the first initial source of this is creatine phosphate. Remember that energy is stored in bonds of molecules. A lot of different things in your cells use ATP, and so in muscles, that energy from ATP is temporarily stored in creatine phosphate. So at rest, energy is stored in the bonds of ATP and is then transferred to creatine to produce creatine phosphate as well as ADP. But then when the muscle is active, that energy that was stored in creatine phosphate is enzymatically transferred back to ADP to produce ATP using creatine kinase. So that is just effective initially. So it only lasts about 15 seconds, but it's a quick way to get ATP. So when muscles initially go from resting to active for the first 15 seconds, creatine phosphate is their source of ATP. After that point, you rely on glycolysis. So remember, glycolysis is that conversion of glucose to pyruvate or pyruvic acid. So it's a slower process and it's anaerobic, so it doesn't rely on oxygen, and it produces small amounts of ATP. It's slower than creatine phosphate, but it's still faster than other processes. So you generate small amounts of ATP and pyruvate. That pyruvate can then have different fates. If you do have oxygen, it can continue through aerobic respiration, but if you don't have oxygen, it might go through lactic acid fermentation. So that is able to recycle different storage molecules. So this is not very efficient. It helps keep you going, but it's usually only sustained for about a minute. So then you have aerobic respiration, which is very impactful. It's a series of reactions. Remember, there's kind of different things that fall into aerobic respiration in which various substrates, so blood glucose, pyruvic acid, different fatty acids, are transformed to heat, carbon dioxide, water, and lots of ATP when you have oxygen using the mitochondria. So remember, muscle fibers have tons of mitochondria in them. They have ways of storing oxygen temporarily to get access to them. So that powers aerobic respiration. And so this accounts for about 95% of the ATP used by muscles. Muscles store that oxygen in myoglobin. So myo, think about muscle. Hemoglobin, think about an oxygen carrying molecule. So myoglobin is a molecule that stores oxygen to power aerobic respiration. And we'll talk a little bit later about exercise physiology and how different types of exercise are associated with greater production of myoglobin. So when muscle metabolism can't sustain contraction, you might end up getting muscle fatigue, in which the muscle can't maintain its strength of contraction or tension. And so the muscle can't contract in response to signals from the nervous system, and it can't produce enough ATP to meet its needs. Muscle fatigue can also be associated with lactic acid fermentation. We also have this idea of oxygen debt or recovery oxygen consumption. That's the amount of oxygen you need to compensate for the ATP that's produced without oxygen during muscle contraction. So when you have intense activity, that increases oxygen debt, and it also results in elevated oxygen use for quite some time. Okay, so getting into nervous system control of muscle tension. Muscle tension, just to define that briefly, is the force generated by the contraction of the muscle or shortening of the sarcomeres. So when you have something being accomplished, that's called an isotonic contraction, but you might also have an isometric contraction in which the load does not move, the sarcomere doesn't shorten. So we distinguish between isotonic and isometric contractions. There's also a relationship between the length of the sarcomere and the muscle tension that's generated, and there's kind of a sweet spot that we'll talk about in just a moment. So this diagram is really helpful. It shows the numbers on the x-axis are kind of length in comparison to length at rest. So 100% is kind of the length at rest, but then you have decreased length and increased length. So it's the percentage of the resting sarcomere length. And then you see the diagrams there to kind of describe what's happening between the thick filaments shown in purple and the thin filaments shown in green. The y-axis is tension. It's measured in the percent of maximum possible tension. So if we look at those little diagrams of the sarcomeres, we see that in some cases the actin and myosin are overlapping, in some cases they're not. So those cross bridges that allow contraction to take place between actin and myosin only form if the thin and thick filaments are actually overlapping. If they're not, then they can't make connections between each other. And if they're connecting too much, they have nowhere to go. So the length of the sarcomere has a direct influence on the force that is generated when the sarcomere shortens. And so there's this kind of sweet spot in that length-tension relationship that we see right about there, from about 80 to 120. So the maximal tension and overlap of binding sites with myosin heads occurs at about 80 to 120% of sarcomere resting length. Below that point, the zone of overlap is reduced and you can't really go anywhere. Above that point, there's no overlap, so you can't even form cross bridges. So in either of these cases, you have less tension being produced. So I also want to define this term twitch. We'll talk about fast twitch and slow twitch. We talk about muscles twitching. Physiologically, twitch means a single contraction of muscle fibers within a given motor unit produced by a single action potential from a motor neuron. So we have this efferent neuron, this motor neuron coming in, sending a single action potential, and that causes a single contraction in the muscle fibers of the motor unit that it's connected to. So this may last for 1 to 100 milliseconds, and this is very different from like your muscles in your eye twitching. You can't really see this single contraction. You need lots of contractions happening in order to actually do work and observe it. But we can quantify this and study it using something called a myogram. So this is a kind of muscle chart where we see different periods and changes in tension over time. So from this single stimulus, from this single action potential and this contraction, I'm going to walk you through the latent period, the contraction period, and the relaxation period. So in phase 1, this latent period, which ranges from about 2 milliseconds, so here it looks like 0 to 5, but it's about 2 milliseconds. This is when the action potential is propagated along the plasma membrane of the muscle fiber, the sarcolemma, and calcium ions are released. So remember, that process actually goes action potential, acetylcholine, voltage-gated channels, and sodium ions, and then calcium ions being released from the sarcoplasmic reticulum. But all of that happens within about 2 milliseconds, that latent period. And then after you have the calcium ions released, you have the contraction period. So those contraction ions bind to troponin. Tropomyosin moves away from those binding sites and unshields them. Those cross bridges between actin and myosin form, and the sarcomeres shorten to peak tension. So you have contraction taking place. This can last anywhere from 10 to 100 milliseconds. This plot, this kind of generic myogram, is showing the contraction period as much shorter than the relaxation period, but it varies depending on the type of muscle fiber, depending on different signals. And so here the relaxation period might also be as short as 10 milliseconds and as long as 100 milliseconds. But this is where tension is decreasing, contraction is stopping, the calcium ions are pumped back into their storage places in the sarcoplasmic reticulum, and cross-bridge cycling stops. So we have the signal being sent in, we have contraction, and then we have relaxation from a single stimulus. So just a few reminders about this idea of individual stimuli versus stimuli coming together. Remember when we talked about the nervous system, we talked about summation of different signals coming together from different dendrites in the motor neuron or in the neuron. We talked about graded potentials and kind of varying degrees of signals. And so we see similar patterns in terms of motor neuron stimulation in muscles. So in order to have a muscle contraction that can actually produce visible work, you need a series of action potentials, not just a single action potential and a single twitch. You need sustained efforts in order to produce work. And so this might be temporal, in which you have a high frequency of impulses, or it could be multiple motor neurons sending impulses spatially, and both of those can affect muscle tension. So this idea of a graded muscle response is the idea that sustained contractions due to a series of action potentials can also be modified by input from the nervous system. That's kind of changing the muscle as a whole, not individual muscle fibers. So when we have summation, we have increasingly stronger combined twitches. Just because of how calcium ions are flowing, it's still kind of responding to the previous one when the next action potential comes in. So every time you have a new stimulus, when you have them in high frequency, those waves summate together, they sum together, you get stronger combined twitches, and then you eventually have what's called tetanus, or a sustained contraction. This process kind of extends in various degrees. It's a little bit more complex than that previous plot. So tetanus is really when relaxation periods between the contractions disappear. You can't see them anymore. So on this myogram, in the really nice clean myogram we looked at earlier, we saw latent, we saw contraction, and then we saw the relaxation being extended outward. Here you have, in this first part of the plot, part A, a single twitch and a really nice myogram with the peak contraction and then relaxation. In part B, you have two action potentials, two signals, and those are summating together. So you have the first one, and then as soon as the second one happens, the peak, the force, is even stronger. And so then it does relax in between. When we have it being extended outward, and we have at this point 4 different action potentials in rapid succession, there's still kind of relaxation period between those. It's increasing still, so you're still having summation, but there is a relaxation period. But by plot D, or like the part D of the plot, you have so many action potentials within such a short period of time that everything sums together and you don't even have a relaxation period. It's just sustained contraction or fused tetanus. You also can have what's called a staircase effect, or trip. So this is what happens when you have a dormant muscle and then you have it start to generate tension again. So it starts off generating about half the tension it maybe once did, but as you receive more signals, it becomes increasingly more efficient. So we're just kind of looking at different myograms and studying those patterns and understanding what's happening here. When we're talking about muscle tone, what this is actually referring to physiologically is firmness due to sustained partial contractions of portions of the skeletal muscle. So which portions those are actually rotate through. You have kind of one motor unit being activated, then another one, so that one of them doesn't tire out. So the muscle as a whole, that organ, is relaxed, but individual motor units within the muscle are contracting. And so this process is important for joint stabilization and posture. A lot of people talk about going to the gym and toning their muscles, or talk about visible muscle tone. Physiologically, muscle tone does refer to firmness, but this is really due to a special type of firmness and a special type of activation that stabilizes our joints and facilitates posture. So if you are not able to accomplish this really, then it's called hypotonia. Hypo meaning low and tonia meaning tone. So it's the absence of these sequential contractions that contribute to muscle tone, and it's usually associated with broad damage to the central nervous system. The converse of this is hypertonia. So hyper means a lot, tonia is tone. This is excessive muscle tone due to damage specifically of upper motor neurons in the central nervous system. And this is associated with excessive reflex response, rigidity, and spasticity. You see this a lot in Parkinson's disease. Okay, so I mentioned this idea of twitch, and you might have heard of fast twitch versus slow twitch muscle fibers. We're going to get in next how we classify those fast twitch and slow twitch fibers. So we do this based on 2 different things. We look at how fast they contract relative to other muscle fibers, and then also how they produce ATP. So we talked about muscle metabolism, we talked about the different methods in which muscle fibers can access ATP. So that's one of the ways we characterize muscle fibers. So there's 3 different types of muscle fibers. There's slow oxidative, or SO. We also call these type 1 slow twitch. So these contract slowly, they're slow twitch. And they are oxidative. They use aerobic respiration, they use oxygen and glucose. There's also fast oxidative, or fast twitch, so specifically type 2A or fast twitch A. These are able to undergo fast contractions, and they're also aerobic. So they primarily use aerobic respiration, they're oxidative, but they are also able to do glycolysis to some extent. And then there's fast glycolytic or FG. These are also called type 2B or fast twitch B. So these are fast contractions. They primarily use anaerobic glycolysis, so they're not oxidative, but they fatigue the most quickly. So it's kind of a balance between fast twitch and so like how fast you can contract versus how quickly you fatigue. So that speed of contraction is actually dependent on the function of that ATP-ASE that's involved in cross-bridge cycling. So remember, it's coming from myosin. So the extent to which it can hydrolyze ATP to produce that cross-bridge cycling is how we classify fibers as fast twitch or slow twitch. And fibers actually do, fast twitch fibers do this ATP-ASE hydrolysis about 2 times faster than slow twitch fibers. This is just a little bit more information about the different types. So type 1, these tend to be smaller muscle fibers. They are slow with low maximum force, but they don't fatigue as quickly. They also have very high oxidative capacity. Remember, they're aerobic, they're oxidative, and low glycolytic capacity. They can't do glycolysis. So these don't produce high tension, but they're super important for maintaining posture, producing isometric contractions, the ones that don't lead to changes in sarcomere length or work actually being done. They are for stabilization of bones and joints. They make small movements that happen very often, but don't require large amounts of energy. So remember, when you're standing or kind of leaning on different things, or just kind of trying to stabilize yourself, there's constant kind of small movements, especially between antagonistic pairs, and slow twitch fibers are important here. Fast twitch, on the other hand, is kind of like in between. It has some oxidative capacity and some glycolytic capacity. These tend to be larger. They have intermediate contraction speed, a higher degree of force, and intermediate rates of fatigue. They also have relatively lower amounts of myoglobin, even though they can go through aerobic ATP production. They're capable of glycolytic or glycolysis. So these, again, are intermediate. These are used for movements with moderate energy requirements, so something like walking rather than something very low energy like posture control or very high energy like sprinting. And these are also kind of like a good balance between tension and fatigue resistance. So then we have type 2B, the other fast twitch, which are very good at glycolysis but can't go through aerobic respiration with oxygen. These tend to be kind of medium in terms of muscle fiber size, but they are the fastest in terms of contraction, and they have a medium degree of force. So they have high amounts of glycogen stored up for glycolysis rather than undergoing aerobic respiration. They don't really need mitochondria or myoglobin because they're not using oxygen-based aerobic respiration anyway. And these are useful for rapid, forceful contractions and very quick, powerful movements, but they fatigue pretty easily. And we can actually see differences between them on histological slides. The slow twitch muscle fibers have much more myoglobin. The fast twitch tend to use glycolysis a little bit more, so they don't have as much myoglobin. So when we're looking at skeletal muscles, remember, a single skeletal muscle is an organ. It has lots of different tissue types, and these skeletal muscles contain a mixture of those different types, so type 1, type 2A, and type 2B muscle fibers. So the one that's present in the highest ratio kind of determines the overall function of the organ, but each motor unit contains just one type. So if there's a motor unit, it's going to have all SO or all FO or all FG. It's not going to be a combination. So that kind of transitions into exercise physiology. And so different types of exercise use different types of muscle fibers. So for example, slow twitch is very useful if you are running a marathon. You don't want to fatigue easily, but you don't need as much extreme power. Whereas if you're a sprinter, you need a very quick start, you need an explosive amount of power, but it's okay if it fatigues easily, so you used fast twitch muscles. So again, kind of with longer running distance, you use slow twitch muscle fibers. For something like powerlifting, you need fast twitch. So when you are doing something like powerlifting or weightlifting or bodybuilding, you might be undergoing something called hypertrophy. So that's when you're adding structural proteins, you're adding sarcomere numbers, so sarcomeres and myofibrils, to increase the diameter of the cell. So you're not actually creating any new muscle cells, no new muscle fibers. That is genetically determined. You can't change that, but you can bulk on the number of sarcomeres and myofibrils. So you can also have atrophy, which is the opposite. That's when the sarcomere and myofibril numbers decrease due to different conditions. So it might be due to a disease pathology, it could be due to age, it could be due to not using the muscle as well, but that's the loss of structural proteins and corresponding decrease in muscle mass. When it is age-related, it's called sarcopenia. So here we have an astronaut who's exercising to prevent atrophy in zero gravity. Remember that a lot of our muscle tone is due to stabilization, and we have to stabilize our bodies a lot to fight against gravity. So when we're not constantly doing that and not putting pressure on these muscles and using them to stand upright and move around, muscles tend to atrophy. This is an example of atrophy after a cast removal. So when you have a cast, you're not using that muscle as much, it atrophies. So the leg on the right is slightly smaller than the leg on the left because of atrophy. And then this is extreme muscle atrophy due to polio or poliomyelitis. So we have a couple different types of exercise broadly. We have aerobic or endurance exercise, so something like running longer periods, and this tends to not cause hypertrophy. It changes other components of the muscle that don't have to do with the filaments. So it doesn't affect myosin or actin filaments. It creates a higher surface area to volume ratio, but doesn't change the filaments. So it doesn't cause hypertrophy, but it also doesn't cause atrophy. It does add more mitochondria for ATP. It encourages angiogenesis, so the formation of capillary networks around fibers. So that's able to bring in oxygen and take out carbon dioxide more effectively. And it also upregulates the production of myoglobin for oxygen storage. Anaerobic exercise or resistance exercise, on the other hand, does change the composition of those muscle fibers. It changes specifically the composition of the proteins. So it causes hypertrophy, you have more myofibrils, you have increased muscle fiber thickness, and there's less cellular changes, but there can be increasing connective tissue. So it doesn't cause angiogenesis, it doesn't cause the formation of more mitochondria, but it might cause an increase in connective tissue to stabilize the muscle. So you should know that aerobic respiration kind of affects non-filament components of the cell. Anaerobic exercise affects the muscle filaments, so like thick and thin, and it also causes hypertrophy, doesn't affect other things as much. Okay, so just to briefly review cardiac muscle, remember that this is referring to heart muscle. It is striated, it is also organized into sarcomeres, but it has shorter fibers, fewer nuclei, and lots of mitochondria and myoglobin. There's connections between the cells in the form of intercalated discs, as well as gap junctions and desmosomes. Those gap junctions allow the cardiac muscle to coordinate electrochemical signals, and the desmosomes keep that cardiac muscle together as it's pumping. So if you look at the GIF on the right, if you didn't have desmosomes and it was pumping, it would be kind of flopping all over the place and not coordinated. In the conduction the cardiac conduction system, you have pacemaker cells. Those are very important for kind of setting the heartbeat. We talked about that with the SA node, and so those are affected by a lot of other things, not just, it's not just dependent on the nervous system, even though it is affected by the autonomic nervous system, but also by other neurotransmitters, different hormones, different drugs. So it's not as dependent on the nervous system as skeletal muscle is, and it's also capable of autorhythmicity. So it's self-excitable. It can influence surrounding cardiac muscle fibers and set its own pace. And remember that action potentials are a little bit different in the heart. So it has extended refractory periods. There's differences with these slow calcium channels. So it's different from skeletal muscle. That cardiac muscle remains contracted longer than skeletal muscle, and it has a much longer refractory period. Okay, so to briefly talk about smooth muscle physiology, smooth muscle again is not dependent on the somatic nervous system. It responds to autonomic nerve impulses and then also hormones and local stimuli. The structure is also quite different from skeletal muscle and cardiac muscle, and it does not have troponin. So if you think back to the beginning of this lecture, I mentioned that all muscle has actin and myosin, but not all muscle has troponin and tropomyosin. So troponin is not present in smooth muscle, and so this regulation is a little bit different. Instead, it has calmodulin, which is a protein, ends in IN, that modulates calcium. So it forms a complex with calcium that activates myosin kinase, and so then you have cycling and binding between myosin and actin. So you should know that calmodulin is regulatory for smooth muscle, that it kind of plays a similar role of regulatory proteins that are found in skeletal muscle. There's also this idea of the stretch relaxation response. Smooth muscles can stretch significantly without developing tension. This is important for the types of organs in which they're found. And then really briefly talking about the development of muscle initially and also regeneration, just a couple different types of cells and a specific process. So myoblasts and satellite cells, as well as parasites for smooth muscles, are all types of stem cells. There are different degrees of stem cells, but the satellite cells can do some level of regeneration. They're mostly for repair. Myoblasts are what fuse together to form a longer myofiber or muscle fiber. And so those nuclei from the myoblasts remain intact. That's why skeletal muscle is multinucleated, because each fiber was originally made up of many myoblasts. And then I also just wanted to finally introduce you to this idea of fibrosis. This is when muscle fibers are damaged and then replaced with scar tissue if the damage is too great to be repaired by satellite cells. So that's it for this lecture. I know it's a little bit longer, but it's just one chapter. So make sure you look over the learning goals and that you're comfortable with them. Next week, there's a quiz scheduled, but it will be posted the following week and will cover material from muscles, urinary system, and homeostasis.